All right, so now we're going to talk about the final stage of protein synthesis, which is termination. And so we have to know what signals are going to terminate in the message, and then how this leads to certain steps that will eventually lead to the dissociation of the ribosome and the release, of course, of the growing protein or peptide. We'll also talk about some antibiotics and toxins that are associated with disrupting protein synthesis. All right, so with respect to our bacteria, the termination codon is going to be in the A site. And so the termination codons is going to be, of course, UAG, UGA, or UAA. This is going to initiate recruitment of what are called release factors. So the release factors will bind. And the termination factors, or these release factors, will be recruited based on the termination codon within the message. So you have a UAG or a UAA in the messenger RNA, and then you'll get release factor 1 that binds. If you have a UGA or a UAA, you'll get release factor 2. And then release factor 3 is going to always be associated with this. So if you get release factor 1 or 2 bind, they will bind to water. They'll add water to the growing chain instead of another amino acid. And our release factor 3 is thought to help to release the ribosomal subunits. So in eukaryotes, there is a single release factor, eukaryote release factor, that recognizes all of the 3 termination codons. So again, you have a termination codon that moves into position A. You get your release factor binding, release factor 1 or release factor 2, depending on which amino acid codon is used. If it's a UAG or a UAA, release factor 1 will bind. If it's a UGA or UAA, release factor 2 can bind. Now for termination in eukaryotes, again, the elongation will continue until you get that last amino acid stop codon by the messenger RNA. Again, the termination codons are the same. And it's important to note that mutations with a transfer RNA anticodon that will allow an amino acid to insert at that termination are going to lead to the cell to undergo apoptosis or die. Now let's just talk briefly about different things that can interact or inhibit our protein synthesis. So we're going to have certain antibiotics and toxins that can inhibit protein synthesis. Antibiotics are going to be specific for bacteria protein synthesis and hopefully not have much of a side effect in us. However, there are some toxins that can affect our protein synthesis. So there's Puromycin. This is made by Streptomyces, and it's going to have a similar structure to the 3' end of our amino acyl tRNA. So this means that it can bind to that A site and is going to participate in peptide bond formation, and it leads to a premature termination of protein synthesis. And so Puromycin will go in there and try and basically prevent any other additional amino acyl tRNAs to enter. And so it's going to block protein synthesis. You don't get a full amino acid peptide, and so your protein does not fold properly or function. Our tetracyclines are going to inhibit protein synthesis in bacteria, and they are going to block, again, that A site, so you're not going to allow more tRNAs to come in. Chloramphenicol is going to also inhibit protein synthesis, and this is by blocking peptidyl transfer. And the nice thing, it has no activity in eukaryotes. Cyclohexamide is going to block the peptidyl transfer of our ribosomes, but not of the 70S bacteria. Streptomycin is going to cause misreading at low concentration and inhibit initiation at high concentrations. So this is the case where you have different levels of toxicity of the same compound. We have some very important toxins. We have Diphtheria Toxin, Diphtheria toxin is a very potent toxin, and it's going to inactivate eukaryote elongation factor 2. So remember, that is the one that brings in the GTP. That's very important for making our peptides. Ricin is from castor beans, and this inactivates the 60S subunit of eukaryote ribosomes. And so, again, you can't make your full ribosomal unit, and you can't make proteins.